Hello and welcome to Nikolay's genetics lessons, and today's problem is analysis of VNTR or STR loci. VNTR stands for the variable number of tandem repeats and STR stands for the short tandem repeats. Has been used in the past to establish paternity in cases where the biological father of a child is in question. Below are the DNA fingerprint of 5 people. In such a case, the child C, mother M, and 3 potential fathers of the child F1, F2, and F3. In this experiment, the total DNA of each individual was cut with a restriction enzyme, run on the 3 gels, and 1000 blotted. Each blot was probed with radioactive probes to different VNTR loci A, B, and C, and the results are shown below. And the first question A, which male or males can be eliminated as biological father of a child C based on this data? So, before I will explain how to solve this problem, let me first explain the theory behind DNA fingerprinting. And this picture would help me to explain what we are doing. So, CODI system were developed by FBI, and in this system we analyze 13 loci on the chromosome number 2, 3, and so on, and also 2 loci on the X and Y chromosome. So if you would count all the loci, you would find that there is not 13 but 15. But the last one, the last two, we analyze just if person is a male or female. So we don't have a variable number of tandem repeats here. We have just a deletion and full version of the allele here. So basically, as you see, all the chromosomes represented here are represented in one copy, but we have 2 copies of the chromosomes, one that we got from the mother side, another from the father side. And variable number of tandem repeats, basically what it does mean, imagine that such a sequence as A, C, G, A, C, G, and A, C, and G. As you see, we have here one repeat that repeats 3 times. Imagine that on the chromosome number 2, at the position, at the locus TP or X, on the chromosome that you got from the father side, you have 3 repeats, and on the chromosome that you got from the mother side, at the same locus, you have 2 repeats of the same repeat A, C, G, A, C, and G. So now we got a unique profile for you for this locus, and we can say that this is 3, 2. So now let's solve a simple problem. If there are 10 alleles for this locus in the general population, and every person has 2 chromosomes, what would be probability of this combination in the population? You may say that we have 10 variants for the first allele, we have also 10 variants for the second alleles, so total number of combinations would equal to 100, and this is going to be a wrong answer. I'm going to use example with not 10 alleles but 5 alleles to demonstrate that we have a number that is going to be much less than 100. Imagine that we have 5 alleles in gene pool. So basically on the chromosome from the mother side we may have variants 1, 2, 3, 4, and 5. And also on the chromosome from the father side, we also may have variants that is going to be also 1, 2, 3, 4, and 5. So here we may have, one second, let me draw a table. So this is going to be 5 on 5, 5 by 5, so 25 cells, 25 combinations, and combinations are going to be as follows: 1 and 1, 1 and 2, 1, 3, 1, 4, 1, 5, 2, 1, 2, 2, 2, 3, 2, 4, 2, 5, 3, 1, 3, 2, 3, 3, 3, 4, 3, 5, and 4, 1, 4, 2, 4, 3, 4, 4, 4, 5, and the last row 5, 1, 5, 2, 5, 3, 5, 4, and 5, 5. So if you think that we have 25 combinations here, this is not true, because combination 2, 1 and 1, 2 is the same combination. So 2 and 1 is the same as 1 and 2. Basically, this tells us that this person, A, has 2 alleles with 2 repeats and with 1 repeat. And person B also has 2 alleles. One is with 1 repeat and another one with 2 repeats. But basically we cannot tell when we run a gel. So imagine that this is a gel. We cannot tell the difference whether, say, allele with 2 repeats are from mother side or from father side. Basically, when we load the gel with this mixture of 2 alleles, and when we run a gel, of course, the band that is going to be at the longest distance would be with smallest number of repeats, so number 1, and the second would be, say, somewhere here with 2 repeats. So it would travel the least distance because molecule is bigger and would move through the gel slower. So basically, as you understand, if we would load a gel here with this mixture and this mixture, we would get the same picture on the gel. So what we have to do, this would be unique combinations that would repeat only once, and the rest, as I said, would be repeated twice. So 2 and 1 and 1 and 2 is the same, 3 and 1 and 1 and 3 is the same, 3 and 2 and 2 and 3 is the same. So we can exclude all these variants here. So what would be our calculations? We had 25 cells, 5 cells are unique, so 25 minus 5 would be 20 cells, and out of 20 cells, half would repeat. So basically we have here 10 plus 5, so 15 unique combinations instead of 25 that probably some of you may predict. So also here we are not going to get 100 unique combinations, but instead, one more time, mentally you can build such a square, such a table, 10 by 10, 10 would be unique, 10 combinations would be unique. The rest, or 90, we have to divide by 2, and we are going to get 45 plus 10 would give us 55 combinations. So basically we can say that 10 by 10 would give us 65 instead of 100. One more time, 65 unique combinations of alleles. If we take into account that there are 10 alleles are present in the gene pool in equal amounts with equal frequency, so in diploid organism we can get 65 unique combinations, unique from the point of view when we load restricted fragments of the DNA, and we are going to get unique combinations here. But once again, 65 here is not equal to 65%. So every time we have to do our calculations. The greater the number, the closer this number would be to 50%. And the smaller number of combinations or alleles in the gene pool, the greater would be deviation from the 50%. One more time, if person A got this allele with 2 repeats from mother side, if person B got this allele also from the mother side but with 1 repeat, and if person A got this allele from father side and person B got different allele from the father side, when we run a gel, these two genotypes would look for us identical. Allele 1 and 2 would separate, but genotype A and B for us would look the same. Now we can return to our problem. I hope now you understand much better what we will see on these gels now. So question A, which male or males can be eliminated as the biological father of a child C based on this data? So M is genotype of the mother. We don't know the size of the alleles. We only can reference to the sizes according to other sizes of the other people that is present here, or the genotypes. So this is genotype of the mother, this is genotype of the child. What we can tell? We can tell that mother couldn't contribute this allele because she doesn't have this allele, but she can contribute to genotype of the child this allele. So mother has this allele and child has this allele. But also probable father number 1 has this allele. So can he contribute this allele to a child? Yes, he can. If a child would have only one band and wouldn't have this band, we can say that mother can contribute this allele and father can contribute the same size allele. Probably child would have this allele on the gel as a more thick band or more bright band, but we see that child has also other allele that cannot be contributed by mother. And by the way, this person number 3 also has the same size allele. So we have to look who is contributor of this allele. And as you see, we can say that person number 2 and 3 can contribute this allele. But what we can tell for sure that person number 1 cannot contribute this allele. So we can exclude him. Let's now move to the gel number 2. One more time. This is genotype of the mother. This is genotype of the child. And mother cannot contribute this allele. She doesn't have this size allele, but she has this size. So we can tell that this allele, this child, got from his mother. And now we are looking who can give this size allele. And as you see, this size allele only can be contributed by person number 2, F2, but person F1 cannot give the size allele to the child, and F3 also cannot give this size allele to the child, so we can exclude them from according to this results. Now let's analyze the last gel. We also call this gel run, and this time this is genotype of the child. One allele can be contributed by the mother. Both mother and child has the same size allele, but mother doesn't have this size of allele, so we have to suggest that this allele are from father side. So who has also such size allele? Person number 1 and person number 2. We can exclude person number 3 because person number 3 doesn't have this size allele. So one more time, the first question, which male or males can be eliminated as the biological father of a child C based on this data? We can eliminate person number 1 and number 3 here, and number 3 here. So we can tell that basically we can eliminate F1 and F3. Question B, which male or males can theoretically be a biological father of a child C based on this data? Only person F2 can be a father of this child because none of the gel eliminated him as a father. He has the same allele for the first locus. One of his alleles for the second locus are also present in his genotype and child genotype, and also one of alleles that he has also present in a child from the gel run number 3 for locus number 3. Question C. Consider that the frequency of each VNTR allele shown below is 1/10 of the general population. What are the chances that the male or males from part B are not the biological father of the child C? So basically, what we have here, we know that this allele have been contributed by the mother. So we are looking for the probability that any of the possible fathers would contribute this allele. So if we would take just a random person from the street, what would be probability that this person would have this size allele? And one more time, frequency of this allele would be 1/10. But because we have two chromosomes, now we have to increase probability. So if we would have only one chromosome, we can say that probability would be 1/10. But because we have two chromosomes that may have two variants of different size alleles, the probability would be 2 out of 10, or 1/10 plus 1/10. So this is probability for the chromosome 1 to have the same size allele as this child, and this is probability for the second chromosome. So all chromosomes goes in pairs. So this is how we got this number, or we also can say that 2/10 is equal to 1/5. So basically we have probability 1/5 here, 1/5 here, and 1/5 here. Now we have to multiply all these independent probabilities, and we are going to get 1/125. This is going to be a probability that if we would take a random person from the street, he would be possible father of this child if we would only take into account 3 loci. If, for example, we would take 1000 or to be more precise 1250 people, there would be 3 people who also can be father of this child according to our calculations. So basically, when we have only 3 loci, we can say that someone cannot be a father. For example, F3 or F1 cannot be a father of the child. So we can exclude someone, but this is not enough exclusion power to include someone, to say that person number 2, F2, is a father of this child. Because if we take, say, 12,000 people, 100 people can be father of this child based on just calculations of 3 loci. So in order to tell for sure that someone is a father of the child, we have to analyze all 13 loci from the CODIS system. Then our exclusion power would be so great that we can say that random person taken from the street have a genetic profile that has only 1 out of 10 billion people to have such a genetic profile, so he could be a father of the child. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.